This question shows cells from the lower part of a transpiring leaf, and we are asked to identify cell B type shown here. So we will write that cell B is a spongy mesophyll cell. The spongy tissue is located just above the lower epidermis and is characterized by many intercellular spaces, numerous chloroplasts, and a round shape, which are typical features of spongy mesophyll cells. This is another question related to leaf tissue, where cells A and B are shown. We know that A represents palisade cells and B represents spongy cells. The question asks for the term used to describe both A and B together, which are the palisade and spongy cells. The answer is, the term used to describe a group of cells like A and B is called mesophyll tissue. Remember, a group of cells is referred to as tissue, so it is important to include the word tissue here. In the next part, the diagram shows an enlarged cell from the above tissue, marked as part A. And we know that this represents a palisade mesophyll cell, and the question asks us to complete the diagram by drawing one chloroplast and 3 other cell components, which we need to label. I'll start by drawing the inner boundary, which represents the cell membrane. Please don't mind as it's not very neat. This is the cell membrane, the inner layer of the plant palisade cell. Next, I can draw a small, round nucleus, which is usually located at one corner of the plant cell. I will shade it, as it stains more deeply and appears darker than the other organelles. Then, I will draw the sap vacuole, a large, central sac-like structure that occupies most of the space in the plant cell. Now, I'll draw a chloroplast, which is small and oval-shaped. It can be located in various positions within the cell. It can be here, 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 even at the sides, typically at the corners, and it appears lighter and smaller than the nucleus. Now we can label the components. I'll label only one chloroplast, as requested in the question. This is the cell membrane. And this is the nucleus. And this is the sap vacuole. This is the cytoplasm. You can label this as well, as it will earn you marks. Chloroplast. Cell membrane. Cytoplasm. Sap vacuole. Nucleus. Label correctly to earn full marks. They are now asking us to draw a line to label the cell in the lower epidermis that contains chloroplasts. To do this, let's scroll up and refer to the diagram of the leaf tissue. In this diagram, we need to identify the cell in the lower epidermis that contains chloroplasts. Be careful to only label the cell found in the lower epidermis. Here, we can see the lower epidermis. The cells in this layer that contain chloroplasts are the guard cells. These 2 cells are the guard cells, and they are the ones that contain chloroplasts. By labeling this correctly, you can easily earn your 1 mark. The question asks about a cell treated with iodine solution, and we need to determine the colors of the cell wall and starch grains after they react with iodine. Iodine primarily stains starch in plant cells, turning it a blue-black color. However, iodine doesn't interact strongly with cellulose in the cell wall, so the wall remains orange-brown or light brown. When iodine is applied, the cell wall generally appears orange-brown or brownish. In contrast, starch grains turn blue-black when exposed to iodine, as iodine binds to the helical structure of starch molecules, producing this distinct coloration. Therefore, the correct answer is C, since the cell wall remains orange-brown and the starch grains turn blue-black after being stained with iodine solution. The question presents the shape of a red blood cell and asks about the advantage of its shape. A red blood cell has a biconcave shape with a central indentation. This shape is essential because it allows the cell to carry oxygen efficiently by increasing its surface area, which is necessary for better diffusion. Thus, option C is the correct answer, as it enhances the surface area for diffusion. Next, the image displays cells from a plant leaf and asks which structure contains a high concentration of magnesium. This is also related to Chapter 6. So, let me point out that option B is the correct answer here, as the palisade mesophyll cells contain more chloroplasts, and therefore, more chlorophyll. Magnesium is a key component of chlorophyll, so in regions with a higher number of chloroplasts, the magnesium concentration will be greater. This question refers to 4 different types of cells: bacterial cell, liver cell, red blood cell, and onion epidermal cell. It asks, which structure is represented by X, based on the options provided. So, option A is the correct answer because X represents the cell membrane, which is present in all of these cells. Option B is incorrect because the cellulose cell wall is only found in plant cells, such as the onion epidermal cell, and is absent in the others. Option C is also incorrect because the nucleus is absent in both bacterial cells and red blood cells. Option D is incorrect as well, since the sap vacuole is present only in the onion epidermal cell and is absent in the others. The diagram depicts a plant cell placed in a concentrated salt solution, indicating that the solution has a higher concentration of solutes, such as salt, compared to the cell's interior. The question asks, which numbered structures are partially permeable? Let's identify the structures first. 1 is the cell wall. It's represented by the double boundary and it is the outermost layer of the cell. 2 is the cell membrane, which has undergone plasmolysis, meaning it has shrunk away from the cell wall. 3 is the cytoplasm, which is the interior of the cell, located inside the cell membrane. And we know that, among these, only the cell membrane is partially permeable. Therefore, the correct answer is option D. The question asks us to compare the structure of a typical virus with that of a typical animal cell. We will write that a virus is significantly smaller than an animal cell and can only be observed under an electron microscope, whereas an animal cell is visible under a light microscope. Both viruses and animal cells contain genetic material, but viruses can have either RNA or DNA, while animal cells contain only DNA. Animal cells are surrounded by a cell membrane, while viruses are enclosed in a protein coat, known as a capsid. Additionally, animal cells have a defined nucleus and membrane-bound organelles within the cytoplasm, whereas viruses lack a nucleus and organelles. We have a question about the importance of cell division in seed germination. We will exclude part A, as it is not relevant to our discussion, and instead focus on the significance of cell division in this process. The process of cell division, specifically mitosis, plays a vital role in seed germination. It enables the growth of the embryo, facilitates tissue differentiation, and ultimately contributes to the development of a mature plant. During mitosis, a single parent cell divides, producing genetically identical daughter cells. This process ensures that the genetic information of the parent is preserved, maintaining consistency in the plant's genetic makeup. Mitosis is essential for seedling growth, as it allows for an increase in cell numbers. This increase provides the necessary protoplasm for the formation of new cell structures. As new cells are produced, they acquire specialized functions that are crucial for overall cell function and development. These newly formed cells then begin to differentiate, becoming specialized for specific roles in the plant. As differentiation continues, the cells form key structures such as the plumule, embryonic shoot, and the radicle, embryonic root. These structures further develop, leading to the formation of a mature plant. The process of cell division through mitosis is thus foundational to the growth and development of the plant, ensuring the proper formation of tissues, organs, and functions required for a fully developed organism. This detailed explanation covers the importance of cell division at various stages of seed germination, from embryo growth to tissue specialization, highlighting the crucial role of mitosis in this process. Next, we have the same question that we've already discussed. Sometimes, exam questions are repeated from past papers. This particular question shows cells from a plant leaf and asks which structure contains a high concentration of magnesium. And we have already discussed that the correct answer is B because the more chloroplasts present, the more chlorophyll there will be, and consequently, a higher concentration of magnesium. In the next question, we need to select the correct functions of the xylem vessels from the given options. Let's break it down. Does the function of xylem vessels include strengthening the roots and stems? Yes, it does, as xylem vessels have tough lignin walls that provide support and help maintain the plant's turgidity. Does xylem also support the shape of the leaf? Yes, it does. However, does it transport sucrose? No, it does not. The transport of sucrose is carried out by the phloem, not the xylem. Therefore, option B is the correct answer, as it accurately reflects all the correct functions of the xylem vessels. This is again a repeated question. It asks about a cell stained with iodine solution, and we need to identify the colors of the cell wall and starch grains after they interact with iodine. When iodine solution stains the cell wall, it generally appears orange-brown or brownish. Iodine primarily stains starch, if present, in plant cells, turning it a blue-black color. However, iodine doesn't react strongly with cellulose in the cell wall, so the wall remains orange-brown or light brown. In contrast, starch grains turn blue-black when treated with iodine, as iodine binds to the helical structure of starch molecules, producing this distinctive coloration. Therefore, the correct answer is C, as the cell wall remains orange-brown, while the starch grains turn blue-black after staining with iodine solution. We have now reached the final question of our past paper. Let's go through it quickly to wrap up this video. The question asks about the adaptations of xylem vessels that enable them to conduct and support the plant. As a reminder, xylem vessels are hollow tubes without any cell content or cytoplasm, and their lignified walls provide strength and support. Therefore, option B is the correct answer, as the key adaptations of xylem vessels are the absence of cytoplasm and the presence of woody cell walls, both of which assist in conduction and support. That concludes our Chapter 1 practice. I hope you enjoyed the learning experience and found it helpful in clearing up any concepts or confusion. Thank you for watching, and I'll see you next time.